Jen Slouch. She is originally from the coastal bayous of Houston, Texas. Jen is a PhD student and NSF graduate research fellow in Dr. Hammer's lab at the University of California, Irvine, Ecology and Evolutionary Biology Department. Her thesis focuses on the native bee communities of wetlands and the factors that shape bee microbiomes. Academically, she is interested in plant pollinator interactions, community ecology, and biodiversity conservation. She aims all of her work to be centered around curiosity-driven science communication through outreach, education, and mentorship and art. So that is Jen. And with that, I will let her take it
Cool.
away
Thanks so
with her
much.
presentation.
Awesome. Hi, everybody. First, I want to say thank you so much for having me here tonight. And thank you very much for awarding the O'Neill grant. The money went a long way toward my research and my project. Today, I'm going to be talking a little bit about one of my chapters for my PhD thesis, which is centered around California's native bees, specifically the bee community and conservation in wetlands. So you already got a little bit of background about me. Year 3 of my PhD, which means I'm about halfway through. So you'll be getting some preliminary results taken for a few more years. And I consider myself a community ecologist, really interested in bees and plants and ants and other things as well. Those have kind of been my main focus professionally. And like Mel mentioned, I'm really in science education through photography and hopefully dabbling in video sometime soon. So I'm going to be talking about native bees today. I really love bees. I think you should too. 90% of flowering plants rely on pollinators. So if you love plants, then you also love bees. There are lots of other types of pollinators besides bees, but they are generally the most efficient pollinators in most of the terrestrial ecosystems around the world. Something that makes bees amazing is there are around 20,000 bee species across the world, and they have these really diverse natural histories that connect them to the environment in really unique ways. Some of these categories are diet breath, so some bees will feed on basically anything they get their hands on. Some bees will only choose one or a few types of flowers. Nest type, some bees nest in the ground, some nest in twigs, some nest in hives like honeybees. Foraging range, bees will have a central nest and some will only forage really close to that nest, like less than a kilometer. Some will forage miles and miles to go look for good plants. Body size, you probably think of honeybees and bumblebees, but most bees are actually quite small, and this affects the way that they interact with the world. And then you also have carpenter bees, on the other hand, which can be larger than your thumb. And then finally, sociality is probably the main thing that you think of when you think about distinguishing bees. Bees run the whole spectrum of truly eusocial, like honeybees and bumblebees that have a hive and a queen, all the way to bees that are solitary, which is actually most bees, where they have a single queen that lays the eggs and raises them all on her own. Because of this amazing diversity, bees are a really useful study group. They're relatively easy to find in habitats. They're relatively easy to identify, at least to the genus level. And they have these really strong connections to the environment through pollination networks, the ecosystem services they provide, and they are then affected by climate change in a way that we can try to measure. And there are around 3,500 native bee species in the United States, and 1,600 of those can be found in California. So if you like bees, this is a really great state to be in. You've probably heard of native bees declining. And this can be for a number of reasons that are kind of interconnected, and many of them are human induced, but this includes habitat loss and degradation, pollution, pesticides, pathogens, climate change, and non-native species. And when you're thinking about how to study the declines in wild bees and how to maybe mitigate that decline, I was trying to pick a study system, and I decided to take a habitat approach and look at wetlands. As I mentioned earlier, I'm from the coastal bayou of Texas. And so when I came to start my PhD in California, wetlands and their destruction were a very familiar problem to me. And California wetlands are fragmented and vulnerable. You've probably seen the statistic that fewer than 10% of California's historic wetlands currently remain. So the next logical step was to look at what type of bees are found in the wetlands. And when I started looking into the research, there was actually almost no literature on what type of bees can be found in wetlands. Bee diversity is highest in the deserts of the United States. So a lot of people go focus there for the high diversity and the fun biodiversity. But baseline surveys are necessary to inform the management strategies for pollinators in any habitat. In the last 30 years, only around 13 published studies have happened in any wetland for the bee communities across the whole US. And there hasn't been previous bee monitoring at most of the wetland sites in California. So the main goal for my project was to establish these repeatable monitoring protocols, which can help us track bee populations over time. So my specific question for my thesis, because this is a lot of, if I studied every threat, then I would have, 6 chapters, and that's just a little too much for 5 years. So I decided to focus specifically on the threat of non-native species, asking the questions, how frequently are native bees foraging on non-native plants? And then how do the non-native plants maybe affect the bee communities? So to kind of look at this with a little bit of ecological theory, if we take the question, how frequently are bees foraging on non-native plants? There's kind of a little bit of theory to back up one hypothesis, which is that the solitary bees will actually forage on non-native plants quite frequently. One of those is optimal foraging theory. So non-native flowers can be very showy and abundant. They're often ornamentals that escape, or the reason that non-native flowers then become invasive is because they're very productive and they do very well in terms of biomass and flower count. And these can be like easier access for native bees. So the bees might actually choose to forage on those, even if that wasn't their historic preference. And in wetland habitats, we have a good idea that most of the bees that are in the wetlands are bees that nest in stems, because in wetlands, the ground is wet. And so there aren't as many ground nesting bees, which tend to be specialists. So if there's more stem nesters in the wetlands, these dominate the wetland sites. They'll forage quite generally on the flowers. And so we can kind of hypothesize that the solitary bees in the wetlands will be foraging on the non-native plants. And if that is the case, the implication for that would be that management recommendations should be made to ensure that you're removing invasive plants outside of when those bees are foraging and feeding, which is not necessarily what they currently do in these sites. For the second question, how might non-native plants be affecting the native bee community composition? A little bit for pollinator networks, they're kind of, the study behind pollinator networks is really complex, and I'm just now beginning to learn how to analyze pollinator networks and study them. But what we do know is that most pollinator networks, so that bee-plant interaction, are quite generalized. So a lot of the species on the bee side and the plant side will have a lot of connecting partners. So pollinators will choose to feed on many flowers, and flowers will choose to attract different types of pollinators. And non-native plants can actually make pollinator networks, pollinator webs, more connected because of those generalist pollinators. But then a downside of that is that the specialist bees that only feed on one type of flower might be outcompeted by a generalist pollinator. So I hypothesized for my system that bee community abundance, so the number of bees, will remain consistent because of all the generalist bees that are in the wetlands, but the richness, so the number of species of bees, will be lower in sites that have more non-native flowers because of that competition with the generalist pollinators. And the implication for this is that we might not lose all of the bees. We might not see like a total loss of bees, but we might lose certain species, those specialists, which will decrease the biodiversity in sites. So that was the theory. And let's dive into the very fun methods that I've been doing for the last 2 years. So I had 8 survey sites across coastal California, 2 in San Diego, 2 in Orange County, 2 in Santa Barbara, 1 up in Santa Cruz, and 1 all the way up in Bodega Bay. And I had a couple of undergrads helping me this year to get the number of surveys that I was able to get. The first year I only had 8, and then this year I think I did 38 surveys thanks to the undergrads, so they were great. The methods that I chose to do were transect surveys. These were 1 kilometer transects that were composed of both passive pan trapping and active netting collection. This allows you to get the broadest types of bees without over collecting. And pan traps are a really common way of collecting bees because they're easier than having somebody wander around with the net. But they do have a tendency to over collect certain types of bees and especially generalists. But the way pan traps work is they're these little spray painted salsa cups that are spray painted 3 different colors, and they're set up in little sets along the transect. I had some on the ground and some that were raised because there's a lot of tall vegetation and a lot of wetland habitats. So by having some on the ground and some higher up, you can get both low flying and higher flying bee species. So these pan traps are set up in the morning. They're left out for 4 hours, filled with a little bit of soapy water. And then after those 4 hours, I go collect any bee that has fallen into the trap. The next day, I come back with a standard bug net, and I go along that same transect, which is broken up into 10 segments. And with my bug net, I set a timer for 10 minutes, and I collect any bee off of a flower that I can find. And I record what flower that bee was on, which gives me that plant pollinator interaction. So you'll be seeing the results of that data later. And over the course of the transect, you end up with 100 minutes of total sampling effort. And this sampling effort is a really important piece of data to collect if you're trying to do standardized monitoring methods. And this one gets more specialized species and it allows you to get that flower interaction, but you typically collect a lot fewer bees with it. So by combining the pan trap data and the net data, you can get a pretty good idea of the bee community in that given time. I also did flower counts because I was interested in how the flower community was changing and shaping the bee community. And so I did bloom observations. So every single flower species blooming, I recorded the number of flower species and the approximate number of flowers per species in view of the transect. So say fewer than 10 flowers, 10 to 100, 100 to 1,000. It's kind of a rough estimate, but because this is a monitoring protocol that we want to be able to apply across lots of different sites, whether you're an undergrad or a volunteer or a student or a classroom, this kind of rough estimate still gives you a decent idea of how abundant certain species were. And because I'm not actually a botanist, I'm just a plant enthusiast, every species was photographed and uploaded to iNaturalist so that my identifications could get corrected. So if you're interested in that iNaturalist project, it's called this, and I can share that with you in the chat later, the California Wetland Bees Microbiome Project 2024. Then I, after I got all of my plant data, I used CalFlora to identify the native status of all of my plants, categorizing them as native, non-native, or invasive. And my preliminary results are this network plot. This is one of the most complicated things I've ever made in the R coding program, and I'm very proud of it, even though it's a bit messy. And so what you're seeing here is bee genera on the left side and plant genera or plant species on the right side. And I know the names are blurry and they're not that important because the bees are not identified yet, and the plant colors are what I want you to focus on. So this is a result of the 41 surveys total from all 8 sites, 2,000 netted bee specimens with their plant connection recorded. And so every single one of those lines is a bee that I collected off of a flower, and that line goes to which flower it was found on. And so then on the right side, there's plants. Blue is for native flowers, tan is for non-native flowers, and brown are for the invasive flowers, so non-native and also invasive. And what I found so far from that is around 1/3 of the bees that I collected with the net were collected foraging on non-native plants. And the percentage of non-native plants did not appear to lower the bee abundance. And I don't have the species count for the bees, so the bee richness, bee diversity yet, because I'm still identifying those, but kind of a preliminary result of this is that more blooms might be better, whether or not they're native or non-native. And so for managing, this might mean that we want to take more care in how we time the removal of these invasive plants because the bees are feeding on them. And then ecologically, I'm curious as to how this affects the bee ecosystem services for native plants. So if bees are now foraging on non-native and invasive species, does that mean that they're not feeding on the native ones as much and they're affecting native plants? And then kind of in turn, what does this mean for the long-term bee fitness? Like, are these non-native plants providing the same nutrients as native plants, or are some of them like cheaper or like not as nutritious? So all of that, stay tuned. I don't have any of those results. These are just the curiosities that have come out of this plot. But I just wanted to end this on a couple of really cool natural history notes that I found when I was out there. So if any of you have ever been up near Santa Barbara, right before you get to Santa Barbara, there's a little tiny wetland called Carpentaria Salt Marsh. And at certain times of the year, this wetland will be covered in Lysthenia glabrata coulteri, which is a Coulter's golden fields, and they are endangered, and they're teeny tiny daisies, and they're beautiful. And I found them to be pollinated by Lasioglossum megastictum, which is a very tiny metallic sweat bee. This site is right next to the highway, and so you just exit and you end up on the site, and then on the other side of the site is the Pacific Ocean. And so the site is less than 1 kilometer squared, and I found these teeny tiny bees nesting within that less than 1 kilometer squared site on the roads, and then pollinating an endangered wetland specialist flower. And so this is really amazing to me because when I started this project, we weren't actually sure that bees were, whether the wetlands even mattered to the bees. It was possible that the bees were living in other places and just kind of showing up at the wetlands to feed on them. But what I found is that the bees are nesting in the wetland and feeding on the wetland plants. And I found nests at every single one of my 8 sites, which is pretty exciting. Another natural history note that kind of goes back to that, like the diversity of the ways that bees make a living in the world, is for the nesting in the marsh, there's a really high proportion of bees that are stem nesters. The most common ones would be Hylaeus, the yellow-faced bee, and Ceratina, which are the small carpenter bees. So if you go to a wetland and you look at the flowers, you'll probably see some small bees that will be one of these 2 genera. And that's really interesting because a lot of management strategies, if they do focus on pollinators, it's on the plant community and not on the nesting needs of the bee. So knowing that there are a lot of stem nesters, we want to prioritize the plants that the stem nesters are nesting in, or maybe more of those, like you've probably seen bee hotels. These would actually be really beneficial in these wetland habitats. The takeaway that I want you to get from all of this information, thank you for listening to the draft of my first chapter, is that wetlands host diverse and valuable bee communities. And I'm really excited to see more of the results that I get as I identify these bee species. So stay tuned for that. And then I would like to thank again the grant from the Orange County California Native Plant Society, the O'Neill grant, also CNSage Audubon and the UC Reserve System, and everybody in my lab and who has helped me design this project. And with that, I think I have a little bit of time for questions, or we might just switch to Matthew, but I'll stop sharing my screen. Yeah.
Awesome. Thank you so much. That was amazing. Does anybody have any questions? You can type that in the chat or say them out loud. I think,
Um,
oh,
Jen, is there any possibility of extending or linking to agricultural farming communities around wetlands? I'm just curious if the bee populations make farming around wetlands more productive, or is there anybody studying that sort of a thing? Because I know agriculture is losing a lot of pollination from bees.
Yes, so there are a couple of people up at UC Davis studying the agricultural systems up in the Central Valley. And so Central Valley is pretty close to the Yolo County wetlands. And there are bees there, but I don't think people are really studying them. If there's a wetland next to an agricultural area, the bees are probably feeding on the agricultural crops there, but um, the more likely issue would be that the honeybees brought over to the agricultural crops would be coming over into the wetlands and competing with the native bees.
Oh,
So that's
yeah,
kind of definitely something worth studying that not a lot of people have looked at, but there are definitely papers on like agriculture and then adjacent wildlands. So yeah.
yeah. Yeah, I was wondering if your research would have any beneficial impact on agriculture either, too, because that's a big problem, that we kind of all need to help solve.
Yeah, no, it's definitely a great path to take in the future. There's a lot of great people studying native bees for agriculture. Yeah,
I hate to say it, I think when it comes to agriculture, that's where the money is.
for sure, for sure.
cool. And then it also looks like Matthew had a question. What are good resources to learn how to recognize bee diversity and identifying bees?
Yes, great question. I'll put them in the chat, but iNaturalist is a pretty great way to see like the most observed bees in an area. So I actually rely on iNaturalist a lot for like whether I'm looking at like a county or like a new site or something. There's a book called Bees in Your Backyard, which is a really great introduction to bee diversity and generally bee families. There's also I'll put a couple of other resources, but that's probably the main one. That's not too daunting. And then, yeah, I think that's my favorite. There's a couple of other books that I'll put in too, but that's the best one. And that's the one that I learned off to get really into the bee taxonomy. And then, yeah, there's a good textbook, The Solitary Bees. Yes.
And then I have a question. Once
Yes.
you're, yeah, but how did you choose your sites? Were you trying to choose more like intact wetlands rather than like degraded areas?
So I was trying to get any site that I could possibly sample on. And the easiest one was since I'm at University of California, Irvine, I have access to the University of California Natural Reserve System. And the first grant that I got that funded my first year was through the reserve system, which meant that I had to choose sites from the reserve system. So I think 6 of them are natural reserves. And then I befriended somebody down at Tawana Slough National Wildlife Refuge. And so that's how I got access to that one. And then IRWD, Audubon Marsh, they're super active, and I messaged them and they were really helpful and supportive of the research for that. Yeah, and then all the rest are reserves. Yeah.
Cool. Does anybody have any more questions before we move on? Okay, well, thank you so much, Jen. That was very interesting.
Thank you.
Okay. Our next speaker is Matthew. Let me get his bio up. But yeah, Matthew, if you want to share your screen and I'll start your bio. So originally from San Francisco, Matthew first became interested in botany while hiking and camping in the Bay Area. Matthew left California to obtain a bachelor's degree in botany from Connecticut College, but spent his summers as an undergrad on vegetation management crews in Rocky Mountain National Park and Sequoia and Kings National Parks. Through these summers, Matthew became convinced of the need for more research in high elevation mountain ecosystems, which he is now carrying out as a master's student at the California Botanic Garden.
Thank you so much, Mel. Let me know if you can hear me and if you can see my screen. Are you able to hear me talking, Mel?
Yes, and we can see
Yes,
your screen.
sorry.
Yes.
Okay, thank you. Well, thank you so much, and thank you to the CNPS Orange County chapter for giving me opportunity to talk about my research. This project was made possible in part from funding from this chapter, and so I'm more than happy to get a chance to talk about progress on this project so far. So I wanted to start just by giving a little bit of background, and as Mel mentioned, before starting this project, I worked for a couple summers with Sequoia and Kings Canyon National Park. And so this is a photo of me with the rare plant crew at Sequoia National Park, and we're surveying a damaged road for rare plants, and we found this rare plant, Erythranthe laciniata. And this plant is actually fairly abundant in some places, but its geographic distribution is fairly narrow, so it is considered rare by CNPS, and ideally the park would like to conserve it. But after I saw this plant for the first time, I started seeing it all over the park, including places that the park's branch of plant ecology wasn't aware of, and a lot of places where rocks and soil from roadwork were actively being dumped on the plants. And so this is just one example from a lot that I could give, where I came to understand that conservation and biodiversity research really require a better understanding of how plants are distributed. And even in a place like California, where we have so many botanists, our understanding of plant distributions are far from complete. And so this is why I decided I want to conduct a floristic study for my master's thesis. I think a lot of you are probably familiar with a floristic study, but just in case not, this is an inventory of all plants in a defined area, simply put. And what I love about these studies is that they're so simple, but they can have some really big impacts. And hopefully that's something that I can demonstrate tonight. Floristic studies can inform taxonomy by documenting new hybrids or sometimes even new species. They can guide hypotheses in biogeography by changing our understanding of how plants are distributed and the types of habitats that they can survive in. Really importantly, floristic studies can affect land management strategies through the documentation of rare or invasive plant populations. And then lastly, also importantly, floristic studies can provide a baseline data set to study change over time, like change in high elevation plant diversity as a result of climate change. And so the area that I decided to do this floristic study in is the McGee Creek watershed. And this is a drainage that's on the eastern slope of the Sierra Nevada in southern Mono County. And so it's about 15 minutes south of the town of Mammoth Lakes, if any of you have ever been there. And then if you've ever driven Highway 395, that's marked in red on this map here, and it goes right through this study site. In total, the drainage is about a little over 26 square miles, and it ranges in elevation from around 6,700 feet down at the edge of Crowley Lake, all the way up to the crest of the Sierra Nevada, which in this drainage is just under 13,000 feet. The land is managed by a variety of landowners, primarily Inyo National Forest, although most of the land north of Highway 395 in this drainage is managed by the Los Angeles Department of Water and Power, and that area is grazed throughout the summer. And so to give you a better understanding of what this site is like, I thought we could take a quick tour of the study site from the lowest to the highest elevations. So down at the lowest elevations, we have this semi-alkaline lakeshore where a number of moisture-loving and salt-tolerant plants are thriving. And just above this, there's a fairly extensive Carex juncus wetland, and a lot of grasses in there too. And this eventually grades into sagebrush scrub. And then up above 8,000 or 9,000 feet, this very abruptly transitions into subalpine forest, dominated by conifers like lodgepole pine, western white pine, western hemlock, and whitebark pine. There's riparian habitat throughout the study site at all elevations, and these support some really interesting plants, including mosses that really thrive in fast-flowing water. There are also meadows at all elevations of various types. There's over a dozen lakes, and these host aquatic plants, which we'll talk a little bit about in a minute. And then my favorite habitat type is the alpine fellfield and boulder field, which is quite extensive in this study area. And this is where some of the toughest plants are braving a really short growing season, the really cold temperatures, high exposure, and high winds. And so one of the reasons that I chose to do work in this study area is that the written botanical history is fairly sparse. But I do want to mention that even though there's not a lot in the written record, people have had knowledge of and an intimate relationship with plants in this area for a long time. This land was and still is the ancestral territory of the Numu people who were forcibly removed from their land. And so this is an artifact that I came across very far off trail. And of course it's still up there, but it was really valuable for me to understand that places that were really difficult for me to get to and that on the surface seemed unexplored were probably intimately known by people not all that far in the distant past. But as far as what we do have in the written record for plants, we have herbarium specimens. We have 247 herbarium records of plants collected where those red dots are shown on the map. And this number really pales in comparison to the watershed just to the northwest, the Convict Creek watershed, where there are over 1,700 herbarium records collected. But it's also not the number of herbarium records that caught my attention, but the fact that 97% of them were collected along the bottom of the drainage where there's either a road or a trail. And iNaturalist observations show the same pattern, where almost all the observations were made along the bottom of the drainage. And the most likely reason for this is just the site is rugged. There are steep slopes rising up on both sides of the trail, and so it takes some effort to get away from the trail. But just because there's been relatively little attention in this area from botanists doesn't mean it's not botanically interesting. And actually one reason that we would expect it to be very botanically interesting is that this site is geologically
I just wanted to, is it frozen? Oh, okay.
Yeah.
Looks like Matthew may have gotten dropped, so we'll wait. Oh,
He's retrying.
Okay,
That's
cool.
Sorry about
Woo!
that. I don't
No,
know
it's all
when it
good.
kicked me off, but let me share my screen again, if that's
You're all
okay.
good. I know, I thought it was just me. I was like, oh no.
Do you remember what I was last talking about?
You were talking about people, botanists not being able to, or not that it's not botanically interesting.
Okay, cool.
Yes.
Okay, you can see my slides?
Yes, I can see the project goals slide.
Great, thank you. Okay, so I talked about that the site is geologically very diverse, and so you could expect that it could also be botanically really diverse. And so what I wanted to achieve through this project, most importantly, I wanted to make a checklist of all plants in the watershed, so both vascular and non-vascular, so including mosses and liverworts. And then second, I wanted to better document the distribution of plants across the range of elevations in geologic substrates, so not just along the trail. And then lastly, I wanted to focus on data of populations on rare or invasive plants in hopes that this data could be useful for land management strategies. And so to achieve these goals, I embarked on 2 summers of fieldwork to do this, where I've been collecting plant specimens. And these specimens get pressed flat in between pieces of newspaper and cardboard, as shown in these pictures. And then along with the physical plant specimens, I also take photos, which eventually end up as iNaturalist observations. And then I take detailed notes on things like when and where the plant was collected, what habitat it was collected in, what species it was growing with, what type of soil, how steep was the slope, what direction was the slope facing, and what the plant looked like when it was alive, and things like that. So this summer I spent a total of 70 days in the field, about a third of which were backpacking. And so what did I find? So as a reminder, here's a map of where collections were historically made in the watershed. And then in the second map, I've added on collections that I made this past summer. So you're able to see that, or you can see that I was able to get to some parts of the watershed where we previously didn't have collection data, but there are also still some gaps on the map, and those are areas that I'd like to target next summer. Out of 1,518 collections, I've only identified a fraction of these, and so unfortunately I don't have numbers like the total number of species that are present in the watershed, but I thought for the rest of the time we could talk about some notable plants that are finding their homes here, and these fall into 3 categories. The first is potential county records, the second is rare plants, and then lastly we'll talk about plants with notable biogeographic patterns. So the first county record that I came across was Erythranthelus ineata, and if you have a good memory, you might remember that this is a plant that I talked about at the beginning of my talk that I first saw in Sequoia National Park. The reason I was surprised to find this in my study area is that, as far as I know, all the occurrences that I'm aware of are on the western side of the slope, and I haven't been able to find collections on the eastern slope and in Mono County. Another potential county record is Potamigene amplifolius, which is only detected in California a handful of times, but is pretty widespread in North America. And this is just a terrific aquatic plant. This was growing in the middle of a deep lake when I saw it, with stems well over 10 feet tall. And I got to swim to collect this plant, and it was by far my favorite plant to collect. And then meanwhile, down in the area that I mentioned is grazed all summer, there are 2 introduced plants, not native to California, that I haven't seen other records for in Mono County, Epilobium leptophyllum and Festuca trachyphylla. There is an iNaturalist observation not very far away of Epilobium leptophyllum in Fish Slough in Mono County. And it's also possible that there are just undigitized collections of these in the area, but it's also possible that these are just relatively recent introductions to the region, and they could represent changes in the flora as a result of human activity. And then lastly, I just couldn't resist talking a little bit about liverworts. So liverworts are very infrequently collected in Mono County, and before this they'd never been collected in the McGee Creek watershed. So here are 7 photos of 7 different liverwort species, and I don't know what these are, but I think I can be pretty sure that at least several of these have not been previously recorded in the area, probably because they're inconspicuous and easily overlooked, and they're kind of inconvenient to identify. The Marchantia polymorpha is kind of the stereotypical liverwort, but I was impressed to see that liverworts are very morphologically diverse, super attractive to look at, and really well worth keeping an eye out for. This summer I came across a lot of populations of rare or threatened plants. Prior to this summer, there are historical records of 3 rare plants, Pinus albicollis, which I guess is threatened instead of rare, the whitebark pine, the meadow lousewort, Pedicularis crenulata, and then Astragalus lemonii, milk vetch. And this summer I came across populations of 30 different rare plant species, most of which were not previously known to grow in the watershed. My hope is that these rare plant records will prove useful because understanding the distribution of rare plants is fundamental for determining conservation strategies. Including in these rare plants, some of my favorites are the Botrychium species, some fern relatives in the family Ophioglycaceae, Botrychium crenulatum, Botrychium ascendens, and Botrychium minganense. And that scale bar in the center photo is in centimeters, so you can see you really have to get on your belly to see these in the tall meadow vegetation. They're really special plants to get to interact with. Also notable was Utricularia minor, notable for being both aquatic and carnivorous. And I think at least some of you are familiar with this genus. But if you're not, Utricularia minor is one of the species that has sac-like bladders attached to its leaves that are held underwater. And when swimming insects get close to the mouth of the bladder and trigger touch-sensitive hairs, the bladder opens quickly, sucking the insect inside, and then closes again, trapping the insect where it gets digested. So also a neat plant that grows in this watershed. And then lastly, I wanted to talk about some plants with some unusual biogeographic patterns. And so to illustrate, I have this map of the range of Salix brachycarpa, variety brachycarpa. And this is a taxon that's fairly common in the Rocky Mountains and also up in Canada and the Arctic, which isn't shown on this map. But for some reason, there are also just a handful of populations also in California. And it turns out there are a few plants that have more or less similar distributions that are common in the Rockies and or the Arctic, but are very rare in California. The Pedicularis crenulata was previously known to grow in the McGee Creek watershed, but Salix brachycarpa, variety brachycarpa, Gabricia Awardees, and Trichophorum pumilum were documented there for the first time this summer. But it's not surprising that these would grow in the McGee Creek area. All 4 of these were previously known to grow just northward in the Convict Creek watershed, which is a watershed dominated by calcareous substrates like marble and limestone. And so this led botanists to believe that one of the reasons these plants are able to grow in California is the unusual geology of this part of the Sierra Nevada. And really interestingly, I can confirm that all the populations of these 4 species that I found this summer, every single population was growing either on marble or limestone, or at least soil with a high pH. And the reason that's interesting is that none of these species are soil specialists outside of California, as far as I know. But this does seem to support the hypothesis that geology is somehow important for explaining the unusual distributions of these species. And I think in general, it follows that appreciated pattern that one of the reasons California is botanically diverse is that it's geologically diverse. And so, of course, this project is not done yet. I still have hundreds of collections from the past summer to identify, and there may be some interesting things in there. I'll be returning to the field in May to continue fieldwork, target areas that I wasn't able to collect in, and try to improve this checklist. And then one way I want to improve the checklist is to try and create more detailed maps of the distribution of invasive plant species. There's some concern that invasive plants will move upslope as climate warms. I haven't been collecting invasive plants every time I see them, but I do mark geographic coordinates in the species name every time I see them. And so my hope is that in creating more detailed maps and a better understanding of the current distribution of invasive plants, that this can help us track where and how fast these are moving in the coming decades. Of course, I haven't been doing this work alone. I'm really grateful for a number of people who helped me in the field this past summer in 2024, and also to the expertise from a number of botanists, including Ann Howell, Ben Carter, Travis Columbus, who's helped a lot and is my advisor, Jim Shivok, and Mike Simpson. And then lastly, this project was made possible with generous support from a number of organizations and individuals, including the CNPS Orange County chapter. This has been such a rewarding project to work on. And so thank you so much for giving me a chance to talk about it and a chance to complete this research. And I'm happy to answer any questions, but I'll leave this screen up for a little bit if anyone wants to scan the QR code.
I was curious, is the area, who owns it? Is it state land? Is it protected? I mean, did you have to get permission to go there, or
Yeah, that's a great
how do you
question.
get access?
So for all areas of land, I had to get permission to collect plant species. The only places that I couldn't go, like go and walk, not collect plant species without permission, was private land. So areas in the community of McGee Creek, which is owned by private individuals, and I'm not collecting in those areas. And then the area owned by the city of Los Angeles, which I got permits to collect in there. But actually, so about your question about what kind of protections there are, I think more than half of the study area is in John Muir Wilderness. So it is wilderness area and has a good amount of protection.
So it's a variety of ownership, huh?
It is, yeah.
Wow. It makes it complicated. Walking
little
one
bit, yeah.
step to there, say, oh, I can't go there.
Yeah. Yeah, it does take some careful looking at the maps to make sure you're collecting where you're supposed to be collecting.
I mean, I assume very few people go there, right?
It's a pretty popular place for backpacking. I had to
It
get
is?
permits to go backpacking, and it was fairly hard to get permits in time. There's some beautiful wildflowers, some
Wow.
great access to the beautiful Eastern Sierra Nevadas.
From my point of view, it's like the middle of nowhere.
Yeah, it is kind of a drive from any major city, but some people want to get out in the middle of nowhere.
Are there campsites there?
There's a Forest Service campground near the trailhead, not too far from the trailhead. And then there's a BLM campground down by Crowley Lake that I've used a lot.
But you go off on your own, right?
Yeah.
Wow. I think I'll just live vicariously through you. I don't get out that much. Wow, it's awesome what you've been doing.
Thank you.
And then it looks like Jen had a question in the chat. She said, do you see a lot of non-native plants in those more remote areas?
Oh, that's a really interesting question. Almost all the non-native plants that I saw, excluding the area that's grazed, that's a different story. Almost all of them were along the trail, except for cheatgrass. Cheatgrass grew up until like maybe 9,000 feet in elevation. I have to check. I haven't sorted through that data yet. And then I did find a specimen of Tragopogon dubius all the way up at 12,000 feet elevation. And I think that was just kind of a waif brought up by some hiker. Probably wouldn't have survived very long up there, but I pulled it out and collected it. It's going in the herbarium.
And then Sunny in the chat said, were there any isoides or Potamogeddon in the higher elevation lakes in the drainage?
Yeah, there are some interesting aquatics there. I think 3 different species of isoides, 2 aquatic and 1 terrestrial. And then that Potamogeddon amplifolius that I mentioned briefly, I think that was the only Potamogeddon species that I saw. But there were a couple of Stoechenia species and then Xanichelia, which are fairly close relatives, I think.
Thank you. And then Mahima also asked, what sort of Eriogonums grow there?
There's a good diversity of perennial Eriogonums. I only have one annual Eriogonum, Eriogonum bailey, Verde bailey. And then there's some of the good perennials include Eriogonum ovalifolium, a couple varieties of that, Eriogonum nudum, Eriogonum incanum. Eriogonum lobbii is probably my favorite though.
And then I had a question, but what made you
Yeah,
choose non-vascular plants? Are you gonna try and like ID all of those yourself?
I wanted to include non-vascular plants, partly because it was something that I wanted to learn about, and since I'm in school, it's a great time to focus on learning as much as I possibly can. Um, and then the other reason is that the reason for conducting a floristic study is me understanding the value of plant data, and I think data on the distribution of non-vascular plants is no less important than vascular plants. Non-vascular plants are pretty easy to collect in comparison to the vascular plants because you just pop it in a little packet and you don't have to spend all this time arranging it nicely and pressing it and making sure it dries in time and making sure it doesn't get wrinkled and stuff like that.
And then Chloe said, was that Tonestis in one of the rare plant photos?
That was Tnessis pearsonii.
And
Yeah,
then I think two
always
more
growing in rock
questions.
crevices.
Yeah. Okay, two more questions, and then I think we'll wrap up. Eileen
Sure.
asked, how long have natives survived there? Hundreds or thousands of years?
That's a good question. I'm sure the composition of native plants has been constantly changing. Um, Interestingly, there are some let me think, maybe not plant fossils. There are some plant fossils from the nearby area from millions of years ago. I'm sure those species aren't there anymore. But yeah, I actually don't know um, how long the set of native plant species that are there now have been there.
Cool. And then the last question is from Bob. He said, you picked years with extraordinary deep snow the previous winter. Was that a problem?
It I mean, it's kind of how it goes in field work. Um, I think last summer there was a good amount of snow. It's good to have enough moisture to to have a lot of things bloom. And then I kind of lucked out that I missed the 2023 winter where um, everything got pushed back like an entire month because the snow was so deep. And so I didn't have to deal with that issue.
Cool. Well, thank you so much, Matthew.
Oh, thank you.
Yeah, and then if you have any other questions, I know that they're happy to answer, Jen and Matthew. So yeah, that's pretty much it. I don't know if Elizabeth or any other board members have anything else to say. But yeah, a round of applause for the two speakers. Oh, I think Noel, did somebody have a hand raised? Or I don't know, just Zoom emojis. But yeah, thank you guys for all coming. Our next meeting will be in person. So that's in January, and our website's pretty up to date. So if you want to see anything with the next few programs or field trips, they're all up for the next few months. Yeah. So have a good night, everyone. We'll see you in person.
Thank you. That was great.
Thank you.
Thank you for coming.